Welcome to this lecture on the pathogenesis of Alzheimer's disease. Today, we are going to explore one of the most pressing challenges in modern neuroscience. We will build our understanding from the ground up, starting with a simple analogy, moving into the exact biological mechanisms, and finally, using mathematics to model how this disease progresses over time. By the end of this session, you will understand exactly how and why this condition develops. Let us start by imagining the human brain as a bustling, highly efficient city. In this city, millions of workers, which represent our neurons, are constantly communicating to keep everything running smoothly. Just like any active city, these workers produce a little bit of normal waste every single day. In a healthy brain, the city's garbage trucks promptly clear this waste away, maintaining a perfect, clean environment. It is crucial to understand this baseline of a healthy, functioning system before we can understand what goes wrong in neurodegeneration. This delicate balance is the foundation of all cognitive function. Now that we understand the healthy city, imagine what happens when this system breaks down. What if the garbage trucks suddenly stopped working? Or the workers started producing a strange, sticky type of trash that simply could not be picked up? Over time, this sticky trash would pile up in the streets, blocking traffic and suffocating the city. Simultaneously, the internal roads within the factories themselves would begin to collapse, trapping the workers inside. This dual failure, both outside in the streets and inside the factories, is exactly what happens in the brain during Alzheimer's disease, leading to a complete network shutdown. This brings us from our city analogy to the formal medical definition. Alzheimer's disease is a progressive, irreversible neurodegenerative disorder. It does not happen overnight. It slowly and relentlessly destroys memory, thinking skills, and eventually the ability to carry out even the simplest daily tasks. When we talk about its pathogenesis, which simply means the biological mechanism that leads to the disease, we are looking at the molecular level. This devastating progression is primarily driven by the accumulation of two specific abnormal protein structures in the brain. Understanding these two proteins is the absolute key to understanding the entire disease. Let us look closely at the first of these two toxic structures, amyloid plaques. These are dense, insoluble clumps formed from a protein fragment called amyloid beta. Crucially, these plaques accumulate in the spaces between the nerve cells, which we call the extracellular space. If you recall our city analogy, these amyloid plaques are exactly like that sticky trash piling up in the streets. Because they sit outside the cells, they physically block the communication pathways between neurons and trigger massive inflammation in the surrounding tissue, which is a primary driver of the disease's progression. While amyloid plaques cause chaos outside the cell, the second structure destroys it from the inside. These are neurofibrillary tangles, which are twisted fibers of a protein called tau that build up intracellularly, meaning inside the cells. In a healthy brain, tau is actually a helpful protein that stabilizes the internal transport system of the neuron, like the internal roads of our factories. But in Alzheimer's, this tau protein becomes abnormally altered and collapses. Without this internal support, the neuron cannot transport nutrients or communicate, ultimately leading to widespread neuronal death and physical shrinkage of the brain. Now that we have defined these proteins, let us visualize them side by side. On the left of this diagram, you see a healthy neuron. Notice the clear extracellular space and the straight, intact microtubules inside the cell, held together by healthy tau proteins. Now, look in the diseased neuron on the right. Outside the cell, you can clearly see the brown clumps of amyloid beta plaques blocking the extracellular space. Inside the cell, the once straight microtubules are collapsing into twisted neurofibrillary tangles. This visual perfectly captures the dual extracellular and intracellular attack that suffocates the neural network. Seeing this cellular destruction brings us to an important question. Why must we understand these exact mechanisms? The answer is that without knowing exactly how and why these toxic proteins accumulate, we cannot stop them. Millions of people suffer from this condition, creating an enormous emotional and financial burden. By studying this specific pathogenesis, scientists have been able to design targeted therapies, like monoclonal antibodies, that specifically aim to clear these amyloid plaques. Furthermore, understanding this allows doctors to use spinal fluid biomarkers, or brain scans, to diagnose the disease years before the first memory loss symptoms even appear. This ability to predict the disease years in advance leads us to our mathematical model. While Alzheimer's is a biological disease, scientists use math to understand how fast these toxic proteins accumulate over time. The formula is A of t equals A0 plus the quantity P minus C multiplied by t. Let us break this down. A of t is the total concentration of amyloid beta at a given time. A0 is the initial baseline concentration you start with. P is the rate at which the brain produces the protein, and C is the rate at which the brain clears it away. Finally, T is the time elapsed in years. Physically, if the production rate P increases or the clearance rate C decreases, the net accumulation grows much faster, pushing the patient closer to the disease threshold. Now that we have our formula, let us see this with a concrete example. We are given a patient with an initial baseline concentration of 10 units. Their brain produces amyloid at 5 units per year, but only clears 3 units per year. We need to find how many years it will take to reach a toxic threshold of 50 units. We substitute our known values into the equation, giving us 50 equals 10 plus the difference of 5 and 3 times t. The difference between production and clearance is 2, meaning 2 units of toxic protein build up every single year. Subtracting the initial 10 units shows us that 40 new units need to accumulate. Dividing 40 by 2 gives us our final answer of 20 years. This means the patient will experience exactly 20 years of silent, gradual protein buildup before symptoms appear. This terrifying 20-year silent period highlights exactly why early detection and treatment are so critical. What if we could intervene? Pharmacological interventions aim to alter this mathematical balance. If scientists can invent a breakthrough drug that boosts the brain's clearance rate, we can effectively slow down the net accumulation of toxic proteins. By closing the gap between production and clearance, the disease progression is severely slowed down. This perfectly illustrates why enhancing amyloid clearance is a major goal in modern neuropharmacology. Let us look at how such a drug changes our mathematical outcome. Let us apply this new drug to the exact same patient from our previous example. We are given the same initial values, but now a drug boosts the clearance rate by 50%. First, we calculate the new clearance rate by adding 50% of 3 to the original 3, giving us a new clearance rate of 4.5 units per year. We substitute this back into our formula. Now, the net accumulation rate is 5 minus 4.5, which is just 0.5 units per year. The toxic protein is now building up at a painfully slow rate. We subtract the initial 10 units to get 40 and divide by 0.5. Our final answer is a remarkable 80 years. By simply boosting clearance, the drug has delayed the disease onset from 20 years to 80 years, likely saving the patient from ever experiencing symptoms. We have covered a tremendous amount of ground today, so let us summarize the key takeaways. First, we learned that the pathogenesis of Alzheimer's disease is fundamentally driven by two toxic structures: extracellular amyloid plaques and intracellular neurofibrillary tau tangles. Second, we saw how this toxic accumulation physically suffocates neurons, disrupting communication and causing cell death. Finally, we used mathematical modeling to prove that the balance between protein production and clearance dictates the speed of the disease. By understanding these mechanisms, you can see exactly how modern medicine is fighting to restore this balance and save the brain. Excellent work today.